So first I want to thank Karina, Philippe, Brett, and Jean-Marc for the kind invitation for this meeting, and also Colette for organizing everything. So I'm going to talk about the structural characterization of the type 6 secretion system. So we haven't heard today about structural biology, but you will see a lot of structure now. So as Melanie told you, the type 6 secretion system is a molecular device that kills all the bacteria. Okay. So as you know, and we have seen that all day long today, there is a number of bacterium-bacterium interaction into the microbiota, and most of these interactions could be beneficial for the bacterial community. So it could be exchange of molecules like chemicals, conjugation, DNA, or chrome sensing, and that leads to the formation of a microbial community. But in addition to this, all these beneficial mechanism, there is a number of antagonistic behaviors. And in this mechanism, there will be delivery of toxin between one predator cell to one prey cell. And that will lead to the lysis of the prey cell, and then there will be a formation of a monospecies community. So the model bacterium we are using in the lab is enteroagglutinative E. coli. It's a pathotype of E. coli, and this strain is able to kill E. coli K12. So I will just show you a movie here. So K12 E. coli is shown here in red, and you will see in this movie, so the movie is something like 50 minutes condensed into 30 seconds, okay? And you will see that after 10 minutes, nothing happened, and then there will be all the red cells that will disappear, okay? So these appear just in something like 50 minutes, okay? So you can read off of all the population of the red cell after 1 hour. And this mechanism is dependent of what we call the type 6 secretion system Melanie just talked about. So in 2012, there was this amazing paper from the Magellan's lab showing that the type 6 secretion systems form this very long tubular structure into the cytoplasm. So this is a Vibrio chorea cell. Okay? And they make this scheme where you can see that this system is composed of 3 substructures, one structure that is anchored to the cell envelope, and one very long, elongated structure, what we call the tail. Okay. So the genes that constitute the type 6 secretion system can be categorized in 2 families. The first family encode genes that are very similar to phage-like proteins, so virus-like proteins. Okay. And as many of you know, there is a number of phages that have been characterized, including what we call contractile bacteriophages. And these phages are composed of the head that contains the DNA, and then there is a tail structure, and the tail is composed of the inner tube, here in black. The inner tube is tipped by what we call the puncturing device, or the spike. Okay? It is assembled on the base plate here, and there is, around the tube, there is this sheath structure. And the sheath is contractile, so it means that it is able to be assembled into an extended conformation and then to contract. Okay. And once it contracts, it will expel the inner tube toward the target cell, and there will be penetration of the tube into the cell. And based on the homology of the type 6 genes with the gene of the contractile phage, it has been proposed that the type 6 will function with a very similar mechanism. And indeed, what Marek and John show in this paper is that the type 6 secretion system could exist into 2 different conformations, one very elongated and thin conformation, and you can see that it seems that there is a lot of density inside the tubular structure, and there is this very short and wide conformation where there is no density inside, so meaning that the inner tube has been propelled outside of the cell. Okay. So based on that, I will just draw the type 6 secretion system like that during all my talk, and you will see that there will be this membrane complex, the base plate complex, and the tubular structure that resembles the tail of bacteriophages. Okay. So first, what we wanted to do in my lab was to define, maybe I will show you that before. So how it works. I just prefer to show you how it works before to show how we determine that. So the first complex to assemble in the cell is what we call the membrane complex here. And then it will recruit the base plate complex, and the tail will be assembled on this membrane complex. Okay. And this is possible to see and to follow the assembly of this tail structure, as Melanie showed you, by fusing the GFP to one of the components of the tail, which is called VIP-A or TSSB in all the other type 6 gene clusters. Okay. So if you do that, you will see that once a prey cell is in contact with a predator, there will be a localization of the type 6 system that will assemble this very long structure. Okay. Then there will be contraction of the sheath, and then 4 seconds later, there is no bacteria anymore. So it has been killed, and that takes something like 4 seconds. Okay. And then the sheath can be recycled by a dedicated ATPase. Yeah. Okay. So if I'm back to the scheme, what I want to show you is what we learned about the membrane complex. So first, we wanted to identify this membrane complex. And to do that, we started with one of the proteins encoding on the gene cluster called tag L. And this tag L protein was obviously a membrane protein. It has 3 transmembrane segments, and it has in the peroplasm domain that is able to bind to the peptidoglycan layer. Okay. So with this protein, we just fuse a histidine tag on this protein, and we try to purify the membrane complex. So when you do that, you can see that in addition to tag L, you can co-purify TSSM, TSSL, and TSSJ. So just to make a very long story short, what we learn about all these proteins is that these proteins are all membrane proteins. TSSJ is a outer membrane lipoprotein, and TSSJ interacts with the peroplasmic domain of TSSM. TSSM is an inner membrane, and it interacts with TSSL. We solve the structure of the cytoplasmic domain of TSSL, and TSSL interact with tag L. Okay. So we solve the structure of all these proteins of the individual component. But what we didn't know at that time was in which order each of these proteins is recruited to the site of assembly, and also we wanted to learn about the whole structure of the membrane complex. To start with the first question, what we did is that we construct fusion, GFP fusion to all of these proteins. And only 2 fusions were functional, GFP to TSSM and GFP to TSSL. So if you look at the localization of these 2 proteins into the cell, you will see that TSSM and TSSL form foci. So there can be up to 8 foci per cell. And these foci here are very static. They don't move in the cell. They stay at the same location all time. Okay. So these foci represent membrane complexes of the type 6 system. So to define in which order each of these proteins is recruited to the site of assembly, we tested the assembly in foci of these proteins in different mutant background. So if you look into TSSJ mutant cells, you will see that the fluorescence of TSSM is diffuse. So it means that in absence of TSSJ, TSSM is unable to find the location where to assemble. Okay. By contrast, if you look into the TSSL mutant strain, TSSM is still able to form foci. So it means that the formation of TSSM foci is dependent on TSSJ, but independent on TSSL. So it means that TSSJ is recruited before TSSM, and TSSL is recruited after TSSM. If we do the same thing with TSSL, we see that the fluorescence of TSSL is diffuse in both J and M backgrounds. So just to make that show that story, so the first protein to be recruited is TSSJ, and then it will recruit TSSM that will recruit TSSL. So to gain insights about the structure of the whole complex, what we did is that we make a double fusion where we fuse the histidine tag to TSSM and the strep tag to TSSJ. So we knew that these locations for the tag were functional, and indeed all the cells here were fully functional. So when we try to purify first by histidine column and then by a strep tag column, we get this 1.7 megadalton complex here. And in this complex, we can find that there was TSSM, TSSL, and TSSJ. So we make some stoichiometry experiments, and we found that there is 10 copies of each of the protein, 10 copies of M, 10 copies of L, and 10 copies of J into the complex. So we move on to do some negative stain electron microscopy in collaboration with Rémi France at the Pasteur Institute. And here is the picture, the image that we get for the membrane complex of the type 6. So here is a top view, and we can see that it is a five-fold symmetry, okay? And here are tilt view until the side view here. Okay, so you can see here that there is a base, there is arches and pillars here. And from this 2D class, we were able to reconstruct the structure of the membrane complex. So I will just show you a movie on that. So this is the membrane complex, the reconstruction. So here is a base. So we know that this part of the complex is located in the cytoplasm, and this part is in the outer membrane. This is a micelle of detergent, where the TSHG lipoprotein is anchored. Okay. So the first, here there is this cytoplasmic base. Here are the transmembrane domain of TSSM and TSSL, and here are the periplasmic domain. And we can find that there is these arches here and the pillars. Okay. So there is a five-fold symmetry, and each of the pillars is composed of 2 monomers of TSSM. Here you can see. One monomer is outside and one monomer is inside. Okay. And then this monomer and this TSSM into the periplasm, they connect to the outer membrane lipoprotein TSHG here that is anchored to the outer membrane here with a micelle of detergent. Okay. And you can see very easily the five-fold symmetry here. Okay. And now if we go to the base of the complex, it is what we see when you are in the cytoplasm, and you can see these 5 bulges here that correspond to dimer of TSSL. Okay. So what we know after that is that this membrane complex is the first complex to be assembled during the assembly of the type 6 Secretion system. And if you do time-lapse experiments here with a double fusion where there is GFP to one of the membrane complex protein, TSSL, and if you fuse the M-cherry to TSSB, so one of the sheath component, you will see that the first complex to be assembled is a membrane complex here, and then there will be recruitment of the sheath protein, and then the sheath will just elongate, in the cell. Okay, so the membrane complex serves as a docking station for the assembly of the tail. So the second complex to be recruited to the type 6 Secretion system is what we call the baseplate complex. So I won't go into details to show you all the data we have generated on the baseplate complex, but we found that this baseplate complex is composed of 4 proteins, VGRG, TSSF, TSHG, and TSSE here. And there will be first assembly of what we call wedges, which are TSSE, F, and G complexes, and 6 of these complexes will just binds to VGRG to form what we call the fully assembled baseplate complex. This baseplate complex will just bind to the membrane complex, and this will be the second step during the biogenesis of the type 6 secretion system. So once it is assembled, there will be assembly of the tail structure. Okay. So as you can see, the assembly of the tail structure needs to have the polymerization of the inner tube in black and the polymerization of the sheath, and these 2 processes should be coordinated. Okay. So the question we ask is how the assembly of the sheath and the assembly of the tube are coordinated. And to do that, we make an hypothesis that one protein in the system will act as a coordinator for that. So if there is a coordinator, it means that this protein should interact with both the sheath and tube subunits. So to define the coordinator, we perform a bacterial two-hybrid assay. So for those of you who are not familiar with the two-hybrid assay, just to make it very short, if 2 proteins interact, the colony will be blue. If the 2 proteins do not interact, the colony will be white. Okay. So we looked at partners of the HCP protein, which is a component of the tube here, and the TSSE protein, which is one of the components of the sheath. And as you can see, only one protein TSSA, so TSSB is the second component of the sheath. Only one protein, TSSA, was able to bind to both the tube and the sheath proteins. So we make the hypothesis that maybe TSSA will coordinate the assembly of the sheath and that of the tube. Okay. So if we look at the dynamic of TSSA, you can see that TSSA, so this is a GFP fusion to TSSA, which is fully functional. You can see that TSSA form a foci very close to the cell periphery, and then this focus will move towards the other part of the cell. And if we make a chemograph of that, we can see that this TSSA molecule is moving at a constant and unidirectional direction, velocity. Yeah. Okay. So TSSA is moving, and this is very reminiscent of the fact that the sheath is going to polymerize, and maybe TSSA is located at the tip of the sheath. So we make a double construct on the chromosome with TSSA fused to GFP and the TSSB fused to M-cherry, so the sheath component. And as you can see here, there will be first positioning of TSSA close to the membrane here, and then there will be recruitment of the sheath subunit to form the sheath, and then the sheath, the polymerization of the sheath, will just push the TSSA molecule towards the opposite side of the cell. Okay, here is a schematic diagram to follow that. So it means that TSSA is able to recruit the sheath protein and then stay at the tip of the sheath during elongation. So we tried to solve the structure, the crystal structure of TSSA, and we didn't succeed to do that. And so we move on and we did some electron microscopy and SAX experiment. And what I show you here is a SAX data showing that TSSA has a starfish-like structure with a central core here and 6 arms. Yeah. So the arms are composed of the N-terminal domain of TSSA, and the 6 C-terminal domain of TSSA are forming this central core here. So obviously it was very clear that we were not able, so that explains why we were not able to solve the structure of TSSA, because these arms here are very labile and they move very easily. So to do that, we just cut the arms here and just try to purify the central core. And with the central core, we solve the structure. So the structure of the central core of TSSA is unique in all the database, and so it has what we call an hexaflexagon. So hexaflexagon, the second name is also kaleidocycle. I don't know if it means something for you. In French, we call that cocotte. Okay? And the cocotte is a piece of paper where you are young, you can just put the finger and just open and close like that. Okay? And in fact, this is exactly what we call hexaflexagon. These are 6 triangles here. 1, 2, 3, 4, 5, 6. And these 6 triangles are bound by a hinge structure here where 2 helices, 1 helix from this triangle and 1 helix from this triangle are connected here. And in fact, these 6 triangles are just connected by hinges like that, and they can just open and close. Okay? So here is a close conformation of TSSA, and TSSA can open with a hinge, and the 6 triangle will open the lumen, and there will be a large channel. And this channel will be the perfect size to accommodate the tube of HCP. So I won't go into all the details for the current model, but what we propose is that the 6 triangles inside TSSA in the central core here will open to leave the space for the passage of 1 HCP protein here. Okay. So we propose a model in which binding of TSSA to the base plate will just recruit the tube component that will interact and pass through the central core, and then the arms will interact with the sheath component, and then they will recruit 1 row of sheath. Okay. And when it is completed, the central core will open again and will allow the access of a new HCP monomer inside the tube, and then there will be recruitment of a new row of sheath, etc., doing for all the assembly of the tail structure. Okay. Once the structure is assembled and it is in contact with the target cell, the structure will contract, it will deliver the toxin into the bacterial cell, it will lyse the cell. Okay. So with that, I will just finish to thank all the people that were involved in this work. So all the structure, and show you all the crystal structure, EM structure, and sac structure have been done by Eric Durand, a very talented postdoc in the lab that is now a permanent researcher. So all the work on the membrane complex have been done by 3 PhD students, Marie-Stéphanie, Raïm, and Laurine, and the work on the assembly of the tail has been done by Yannick and Raïm. Laure and Nicolas are working on the effectors. I didn't talk about that today. And Maxence is involved in a new project in the lab. And I want also to thank all our collaborators, Rémi Franz at the Pasteur Institute, Christian Cambillot at the FMB, Tamino and Léon Espinoza at the LCB in Marseille for fluorescence microscopy, and of course, all the funding agencies. So thank you very much.